everyone. This is Rachel Jerkins, and I am here to teach you about apoptosis in human leukemia, or HL60 cells. Somatic cells divide to grow and repair, and this division is regulated by controlled cell death, otherwise known as apoptosis. Sometimes, however, the cell continuously divides out of control, and this has the potential to cause cancer. The cancer that is focused on in this study is leukemia. Leukemia impacts the lymphatic system, bone marrow, and other tissues that are used to form blood in the body. Patients with leukemia produce abnormal white blood cells that cannot function properly, and this results in the decreased ability to fight infections. The HL60 cell line has been used in laboratory research to find different treatments for leukemia. It was established from a 36-year-old woman with acute myeloid leukemia in the year 1977. Treatments vary from patient to patient. Patients with leukemia that is growing slowly may undergo treatment that involves monitoring. On the other hand, patients with fast-growing leukemia may undergo chemotherapy that is typically followed by radiation therapy or stem cell transplant. Both chemotherapy and radiation therapy are both used to damage the DNA of cancerous cells by breaking down the single or double strands. The purpose of this experiment was to find a plant source to extract polyphenols from in order to induce apoptosis in the HL60 cells to prevent the spread of cancer throughout the body. I chose to use starfruit as my plant source because literature references show that it has been successful in inducing apoptosis in breast cancer cells, but it has not yet been tested in the HL60 cell line before. 3 assays were performed to gauge how well the starfruit extract worked on inducing apoptosis. These assays were the viability assay, the morphology assay, and the comet assay. The first assay, or the viability assay, is used to compare the controls with the treatment from the starfruit extract. This table breaks down the controls used and their absorbance values. The controls included 2 wells with media alone, 2 wells with cells alone, 2 wells with cells and 70% ethanol, and 2 wells with cells and camptothecin. These absorbance values were compared with the 2 wells that contained cells and the starfruit extract. The average value between the 2 wells of each treatment type were calculated. Standard deviation of the 2 values for a given treatment was also calculated. T-tests were performed between the absorbance values of media with the cells versus 70% ethanol with the cells, media with the cells versus camptothecin with the cells, and lastly, media with the cells versus the starfruit extract with the cells. This bar graph shows how both the camptothecin, which is a known inducer of apoptosis in HL60 cells, and the starfruit extract both had a p-value below 0.005. This indicates that they were significantly different from the controls and that they were both viable for affecting the growth of HL60 cells. The next assay was the morphology assay. The point of this assay is to be able to visualize the phosphatidylserine flip from the inner leaflet to the outer leaflet of the plasma membrane that is caused by enzymes called flipases. Any cell shrinkage, condensation of the nucleus, or blubbing of the membrane caused by enzymes called caspases in the cell may also be viewed through this assay if the plant extract was capable of inducing apoptosis. To start this assay, 20 microliters of the starfruit extract was put into a well that contained HL60 cells, and the plate was incubated for 24 hours. After the 24 hours was up, 10 microliters of cyto16 dye that is used for visualizing the blubbing of the membrane and cell shrinkage was added. 5 microliters of a chemical called annexin 5 was added because it can bind to phosphatidylserine, and it was conjugated with an orange dye called Alexafluor 555 that helps visualize the flipping of the phosphatidylserine. After incubating the plate for 30 minutes, the plate was taken to a fluorescence microscope. The green cyto16 dye showed that the starfruit extract started to induce apoptosis. It is represented here by the 2 cells that started shrinking. The Alexafluor 555 shows how phosphatidylserine began flipping in the HL60 cell. The same assay was repeated, but this time the extract was placed 48 hours before viewing the cells under the fluorescence microscope, because the 24-hour treatment only showed the beginning signs of apoptosis. The 48-hour treatment worked better because more HL60 cells shrunk and more phosphatidyl shrinking was noticeable. Here are 3 of multiple cells that shrunk, and here is a cell that underwent a phosphatidylserine flip. The final assay, called a COMET assay, was used to see if the extract was capable of damaging the HL60 cells in DNA. The starfruit extract treated cells were incubated 48 hours prior to viewing the cells on a slide under the fluorescence microscope. Green dye was used to see if comet heads or tails would form. Several comet tails were visible under the microscope, meaning that the starfruit extract damaged the DNA. The tail is caused by the quick mitigation of the negatively charged DNA towards the positive anode during electrophoresis. We know that the DNA was successfully damaged because healthy DNA would have formed a comet head because it migrates slower to the anode. Here we learned that the starfruit extract had a significant difference from the controls in the viability assay. It caused cell shrinkage and phosphatidylserine flippage through the morphology assay, and it damaged DNA of HL60 cells as represented by the tails in the COMET assay. These 3 assays suggest that the extract from starfruit was successful in inducing apoptosis in HL60 cells. However, it is likely that several polyphenols were extracted from the starfruit. In the future, the components of the extract should be separated to determine which polyphenol was responsible for inducing apoptosis by purifying the active component. After this, the ability of that polyphenol to induce apoptosis could be tested in vivo. Xenograft mice, or mice with implanted HL60 cells, could be studied to see the effects of the starfruit extract before the treatment is tried in humans with leukemia.